enhancing the drug discovery process, Bayesian inference for the analysis and comparison of dose-response experiments. Please.
Hello? Yes? Okay, so hi everyone. Thank you for being here. My name is Caroline Labelle. I'm currently a PhD student in bioinformatics in the Lemieux lab at ARIC. And today I'll be presenting how we can use Bayesian inference to analyze and compare dose-response experiment. I'll start the presentation. Do I have the... It's not there? Sorry. We don't have the cursor? Okay. So I'll start the presentation by explaining what are dose response, how we currently analyze them, and what are some of the shortcomings of this approach. And then I'll present our... What? But do I have the course? No, okay. So I'll then present, our Bayesian methodology, and then present some example. Thank you. Some example. Does it work? How does it work?
Sorry.
Which one? Oh, the red one. Sorry for all that. Red one. Yes, okay. So I'll present our Bayesian methodology and then also some application of this approach. So as the name indicates it, dose-response experiment aim at studying the effect of a specific concentration of a given compound to the response it will generate in cells. We can test for many compounds for a single dose, in which case we will then try to identify the compounds that seems to generate the highest response. We can also test for a gradient concentration, in which case we will study then the global dose-response relationship. And finally, we can also study the synergy relationship by doing combination of compound and or concentration. For the moment, we are mainly interested in the analysis of gradient concentration assays. And we want to do this kind of experiment in the hope of characterizing compound efficacy. So this is normally done by analyzing multiple compound on a multi-well plate. So for example, here, each column is compound specific. And then in each wells, we'll have a number of cells and then a specific concentration of a given compound, and the response will be assessed through some viability assays. And we are actually interested in analyzing this list of responses. So the list of responses are actually real numbers, and we'll try to characterize compound efficacy by, normally we do what we call nonlinear regression using algorithm such as the Markov-Levenberg. And what this algorithm does is for a given mathematical model that we deemed best represent the response of our cells, we'll try to infer the free parameters of that model such that the differences between the data point and the estimated responses is minimized. So in the case of dose response experiment, we usually use the log logistic model, which has 4 free parameters. So once we apply nonlinear regression, we will get an estimation for the IC50, which correspond to the inflection point of the curves, which can also be interpreted as the concentration that will result in a response that is halfway to the maximal response. Then we also have the max and min response, which are the upper and lower bound of the curve, so they are asymptotical values. And finally, we have the slope which is the slope of the curves, and will indicate the speed of the shift between our two plateaus. And from these metrics, we can infer what we call a dose-response curve. So this is all the information that we currently use to characterize a compound efficacy. So let's say that we had an experimental data set, and we wanted to see if that compound is effective on certain cells. So if we did the nonlinear regression, we would get an IC50 values. And if we were to base our conclusions solely on this value, we would conclude that, yes, this compound seems to be somewhat effective. So we're quite happy with that. But if we push a little bit further our analysis, we actually see that the maximal value is quite very, very, very low. So only 15% inhibition of cell growth inhibition. And if we push even further our analysis and look directly at the curve, we would conclude that this compound is simply inactive. So from one single set of results, we were able to conclude very different conclusion, which is not that great, because even though we do actually consider most of the time all of these efficacy metrics, we give a lot of importance to the IC50 values, and as we just saw, it could bias our conclusion. So what we should do is, when we analyze those metrics, we should consider their uncertainty, giving the experimental data set analyzed. But this is not always trivial to do. So we ask ourselves, how can we characterize data noise and assess the uncertainty of efficacy metric? And then when we looked at the curve, we said that this compound would most likely be inactive, but this is mainly based on what we think a good curve should look like. So we then asked also ourselves, how can we implement the notion of intuition into our mathematical reasoning? To answer these questions, we turn to the Bayes theorem, which allow us to infer a posterior distribution of some metrics M, giving some observation O, by multiplying the likelihood of the observation, giving some metrics, giving some values metric M, and then some prior information on M. So the prior are in the form of distribution, and they actually represent what we think the metric should take as value before seeing any kind of data. So in the case of our, in the context of our dose response experiment, M would be all of our efficacy metrics, and then O would be the dose response data set. And what's interesting with this little equation is that it actually answers our initial questions. So by inferring the standard deviation in our likelihood relation here, we are actually characterizing data noise. By using a prior distribution to compute our posterior, we are actually implementing our intuition into our analysis. And finally, by analyzing posterior distribution instead of single point value, we could easily calculate metrics of uncertainty such as confidence interval. So if we apply this approach to experimental data set, we get the dose response curves, which is actually the median dose response curve with some confidence interval. And as I said, each metric would now be represented in the form of a distribution. And we can also, from this distribution, we can derive a median value as reference of the metric for each metric efficacy. And then we can also calculate confidence intervals surrounding those median values. And the inference itself is done using Markov chain Monte Carlo algorithm. So how we do it is giving our experimental data set and our prior distribution. We will iteratively sample values for each of our efficacy metrics, and these values are either rejected or accepted and added to the posterior, which grows as the algorithm goes on. And the values are accepted with a probability that is given by the ratio of likelihood of the newly sampled value and the last accepted value. So in the end, we get those posterior distribution. If we compare the conclusion from the nonlinear regression and our Bayesian approach, we see that the final conclusion are the same, but with our Bayesian approach, we can go there in a more straightforward fashion. So if we look at the IC50, we see that it's very hard to pinpoint the value that the IC50 would take, but we see that the distribution mainly take very, very high value. So we could say that this compound is probably not effective compared to the nonlinear regression, where we would only look at the IC50, it would have a somewhat satisfactory value. Something else that's very interesting here is if we zoom in on the experimental concentration range, we see that the curve is flat there, and this is what we would expect given our initial data set. And another thing to notice is that we see that the median curve actually goes all the way to our optimal maximal value, which from the data, we know that it's probably not the case. But the confidence interval surrounding that region is very high. And this is a very good example of the role played by our prior distribution. So when the data is insufficient, the inference will actually fall back on the prior distribution. So we are, in that case, considering both observed and unobserved data. And this notion of prior and giving some information regarding the parameter is not new to this kind of analysis. We already do this, but in a very crude way when we actually fix parameters of our models. For example, here we fix the max and the min value to 100 and 0 respectively. So in our paper, we argue that we should be using prior that we define as less informative in the sense that we do not want the prior to outweigh the experimental data. We want the prior to be there as a safety net when the data are not sufficient enough. So we might as well be interested in comparing two compounds, not only comparing analyzing a single compound. And in that case, we can expand our Bayesian approach. So here we are actually calculating the posterior distribution of the differences between the values of two curves for a single matrix. And from this distribution, we can actually calculate the probability of each compound of having the highest or lowest value for this specific metric. So for example, here, we're not able to say that one compound is actually significantly more potent than the other one, but we see that the red compound has a probability of having a smaller IC50, which is good, of 75% compared to the blue compound. And then if we look at the max response, we see that, again, the red compound has a probability of having the highest maximal response of 90%. And if we go to the slope, then the slope, the red slope, has a probability of being steeper than the blue slope of almost 100%. So these kind of analysis can help us to identify the compound that would be interesting to further analyze in subsequent experiment. All of this is very nice, but for our friends, the medicinal chemists and the biologists, it's not always trivial to implement the Bayes theorem with an MCMC. So we actually implemented a web interface called BIDRA for Bayesian Inference for Dose Response Analysis. And this interface allows a user to either do a single compound analysis or a two-compound comparison. The data sets are uploaded in a CSV format. The priors can be the default one, which are the same as the one we define in our paper, or they can also be user-defined. And the results are outputted in the form of informative graphs as well as tables. So we get, as you can see, the graph generated here looks a lot like the graph that I've presented so far. So all the graphs that are in this presentation and in the paper were obtained on our web interface. So we've now created a tool for biologists and medicinal chemists to analyze and compare dose-response experiment while characterizing data noise and the uncertainty of the efficacy metrics, and by also implementing their intuition into the computation. If we were interested in comparing two compounds, we might as well be interested in analyzing the entire plate, in which case we would want to rank the compound in terms of their efficacy. And to do so, this is work in progress. So to do so, here we have 10 synthetic curves that varies in their IC50 values, and we're asking ourselves which one would be the top most potent compounds. So to do so, we actually calculated the posterior, the rank posterior of each compound, and this is what it's represented here. So each row is the rank posterior of a given compound, and for instance, yeah. Compound 1 has a very low probability of having the highest compound, and compound 10 actually has a high probability. And since we're analyzing distribution, we do have those uncertainty region, which could be due to noise in the data, or also could simply be due because the distribution of two compounds are very alike for this metrics. So in this example, we could see that the compound 1, 2, 3 are actually, could be in the top 3 most potent compounds. And to put everything that I've been telling you so far into perspective, I'll tell you a little bit more about the story of compound M. So some of our collaborators back in Montreal actually identify a compound of interest, compound M, that had promising effect in inhibiting cell growth in a certain disease. So they then synthesized a bunch of analogs that were based on this initial compound M. They did dose-response experiments on those analogs, and then they gave us the result, and we were left asking ourselves which analogs would be the top most potent, and then also is there or are there any analogs that could actually be more potent than our initial compound M. To answer these questions, we did our posterior ranking approach, and we actually found 4 analogs here that had a probability of being in the top 5 most potent that was higher than 95%. And what's interesting is that our initial compound M here is actually very far from being in that list, which would suggest that they are indeed compounds that are more potent than our initial compound M. And this is what we actually see when we do an individual comparison of each analogs to our compound M. We see here that the IC50 differences distribution are all significant. So these would be 4 analogs that we would be interested in further analyzing to see if they are indeed more potent or more effective in our, at inhibiting cell growth in this disease. So this is just a snapshot of what Bayesian inference can do for us when we analyze those responses. And we think that it would be beneficiary to actually try to expand this approach to single dose and synergy assays. I would like to thank all the members of the Lemieuse Lab, especially Kameo Samishima, and all the members of the Lucigen team, our collaborators, and a big thank you to Patrick Gendron, Geneviève Boucher, and Anne Marigny. Thank you.
Hi.
Hi.
Really lovely to hear you describing it. I like the approach. But you have a fundamental assumption here that your comparator is the maximum that you get with that specific compound. And if the ceiling for that compound is 13% cell kill, then you might get an exaggerated IC50 while it's actually ineffective. But in real drug discovery, nobody does that.
Nobody does.
So in real drug discovery, what we have is we have control fields where you have, for example, equal concentrations of, say, DMSO, if that's the typical thing. And then you're comparing how much are you killing the cells in comparison with the equal concentration of DMSO. So your ceiling is never the top ceiling for that. So how does this approach compare when you look at it in real data, not in this kind of weird...
but these are real data also. These are experimental
but
data.
nobody really calculates IC50s like that. I mean, no one does that. So what people do is you compare it to the control.
Yes, but here the data are actually normalized, giving the control, right?
Yeah, but your IC50 here, it's basically the concentration needed to kill half of the maximal killing that you can reach.
So could actually,
usually what they, sorry if I
Yeah,
interrupted, but
sorry.
usually what they do, they compute the IC50 as the concentration needed to kill half
Yeah.
of the cell in the plate. And
Yes, I
if you
understand
have
the
the same
question.
range of tested concentration, then the compounds are comparable across each other, right?
Yes. So once you have actually the 4 metrics that I've presented here, you could derive easily for the Y50 or the EC50, that's what you're talking about. You could derive a bunch of other metrics from this approach and get the same distribution about it. The only two...
Nobody is disputing the metrics and the maths. We're disputing that it's a wonderful solution to a problem that
doesn't exist. Okay.
It's beautiful.
Yeah.
I like it. But it really, that problem
Okay.
doesn't exist. We
Okay.
never had that real drug discovery.
Perfect. I would like to...
Yeah, I mean, we can take it offside, but I mean, it really is a wonderful solution to a problem
That doesn't exist.
because nobody should be doing drug discovery. Stop now.
Sorry, if it's about killing a cell, I agree, but there are a lot of cells, for example, capital cells which do not have a control, and there you have to rely on such matter.
You should never do an experiment without a control.
No, it's not about without control. I mean, if you say in case you have a full agonist and then you want to find a partial agonist, then such an equation would be useful. Also, for example, in drug discovery, we often have this, also on drug discovery, you have actually what I found like interesting, for example, you have very phenotypic assays, where actually you cannot have a genetic
Exactly.
or for example, like a real control. You have a phenotype, say the nuclear area is reduced. In that case, actually, this can be very useful.
So,
I see what you mean, but I think it's really depends on the context they are talking about.
so
I think the title maybe is misleading that it's not universally used in drug discovery, but certain circumstances, I do think it's useful.
So I guess the
Good.
argument or questions from this side is specifically is coming in context of cancer research or cancer cell lines, where you have to have a control. Without that, it doesn't make any
Yeah.
sense. And years of experience, people have done these experiments, and reproducibility in this field have taught us that you need good control, first
Yes.
thing. And second thing, I'm wondering, I had a similar question, but I'm moving a little bit ahead and asking you, have you compared it with GR50 metrics, your approach with these GR50 approach or DSS approach, other matrices that are used also?
We did. Some of these approach actually need that you have a negative and positive control, which is not always the case when you, with our collaborators, when they develop the actual protocols of their screens. They don't know.
You should not do science if you don't have controls. Stop now. Become an artist. Honestly,
And-
it's a fundamental of science. You need
Yes.
control,
Control, yeah.
otherwise you could just make anything up.
So another straightforward question, I'm a little bit lost in this point, that most of the time our problem is that we can't compute IC50 because
Yeah.
the curves are either too flat, and it's,
Yeah.
what do you do in those cases, for example?
When the curves are too flat,
Yeah, you can't compute the IC50.
too flat in the
Yeah.
sense that it would look like a parabolic curve? Or?
Yeah, it goes flat or just no
Well,
effect at
you
all.
would infer an IC50 that is actually out of the range that you've observed. And this is what this approach actually does, is that you can have this, you can infer this value and then have some confidence interval around it because the data is not available to actually infer specifically this value.
So, I mean, I can say one can discuss all this stuff about controls. We published the Cell Reports paper in 2017 where we developed a Bayesian framework using Monte Carlo simulation also for drug combinations.
Mm-hmm.
And I can state that throughout that study, all the controls were done as they're supposed to, and we still needed this kind of framework. So I'm not completely sure that the critique being presented here is actually relevant. At least in our hands, doing things correctly with controls, we still needed Bayesian frameworks for doing. This was time stacker study, so we wanted to time stacker the 2 compounds in a full FDA oncology matrix. And to develop this, you had to develop Bayesian frameworks and go way beyond what classic pharmacokinetics can handle. So that's more a comment than an...
Okay, thank you.
Thank you very much. Maybe
Thank you.
let's continue the discussion.
Yeah, during lunch.
Yeah. And maybe like 3 speakers, they did not uploaded the presentations. I would really appreciate. And And one more thing, like there is these posters. Could you please review and vote these give the top 3 posters?